Mitochondrial damage is another form of specific damage. Mitochondria are essential in cell functioning since after glycolysis, pyruvate converts to acetyl coenzyme A that enters mitochondria and by means of the Krebs cycle in the electron transport chain leads to an abnormal amount of ATP production to such an extent that for each molecule of glucose, 34 to 36 molecules of ATP are produced. Mitochondria may be damaged by direct causes like hypoxia or some toxins, or indirect causes as for example oxidative stress, intracellular calcium increase, and phospholipid degradation. Consequently, the mitochondrial membrane loses its potential, thus stopping its respiration processes. ATP production decreases or even stops. But the most severe damage occurs when forming the so-called transition pore on the mitochondrial membrane. Transition pores connect the inside of the mitochondrion with the outside of the cell, and as we're going to see during our lessons on cell death, this leads to severe consequences. The lack of calcium homeostasis is another form of cell damage. In physiological conditions, calcium is extremely more concentrated outside the cell. This cytoplasmatic calcium increase can be observed following hypoxia or ingestion of some toxins. As we have already said, calcium is normally highly concentrated outside the cell. When calcium increases inside the cell, it is usually due to the calcium released by the smooth endoplasmatic reticulum or by mitochondria, and only as a last resort eventually coming from the outside, since it is very difficult to control. The increase in cytoplasmatic calcium can be observed either due to ATPase hyperactivity of calcium carrying it from the smooth endoplasmatic reticulum, or through the mitochondrial carrier, which is a calcium-hydrogen exchanger. This exchanger normally expels H+ ions from the intermembrane space into the cytoplasm and then carries in calcium. However, when the proton gradient is dissipated, in other words, when there's mitochondrial damage, this exchanger does the opposite, thus filling up the cytoplasm with calcium. Proteins releasing calcium give me another way to increase cytoplasmic calcium. Many proteins are normally complex with calcium, but cytoplasm acidification decreases the calcium protein affinity. Finally, calcium may enter the cell following a membrane rupture. Among the most common consequences of cytoplasmic calcium increase, we can find altered protein function due to protease activation, ruptured membrane due to calcium-dependent phospholipase activation, rupture of DNA due to calcium-dependent endonucleases, apoptosis following caspases activation, and opening of mitochondrial membrane transition pores. Finally, an easy to understand but severe form of damage is that of the membrane damage. This damage can be mediated by either hypoxia or toxic agents. Cells losing energy usually lead to incorrect management of osmotic gradients and therefore to cell swelling. Moreover, this phenomenon is linked to bacterial toxins directly inhibiting membrane pumps, to pore formation by complement, and to physical and chemical agents damaging membrane transport proteins. But let's talk about chemical damage, a slightly different damage compared to the others. Chemical damage may be direct or indirect. Direct chemical damage is caused by the direct toxic action of toxins, while indirect chemical damage occurs with toxic agents metabolized by human bodies, and especially in cytochrome P450 mixed function oxidase system, and metabolites act as toxins. Another example of direct chemical damage is that of mercury chloride that binds and forms adducts with a sodium-potassium pump, blocking its function and causing cell swelling. Potassium cyanide instead directly acts on mitochondria, where it binds and stops respiratory complex 4, creating a complex with a heme group. Cyanide-potassium damage also affects the hemoglobin ability to bind oxygen. The examples of indirect chemical damage are slightly more complex. An example is that of carbon tetrachloride that is metabolized in carbon trichloride radical and is capable of doing all the negative actions that radicals do. Another example is that of paracetamol, an antipyretic drug that is converted into a radical form, NaPQI, and acetyl-P-benzoquinone imine. This radical may lead to liver damage and fulminant liver impairment after administering 6 grams of paracetamol. The simplest way to contrast intoxication is through glutathione administration. However, remember that only 5% of paracetamol is converted into this metabolite, since the rest undergoes glucuronidation and is excreted via the bile. However, by increasing a lot of doses of paracetamol, the amount of NAPQI will increase up to toxicity. This threshold can be reduced in subjects with glucuronidation metabolic disorders. Thank you for watching. This video was created by School of Biomedical Sciences, Agorà. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.